Terrific. Thank you very much. Pleasure to be here amongst so many old colleagues and friends, as well as many, many new and young ones that I hope will become colleagues and friends for years to come. I would like to tell you a little bit of the work we've been doing with the SHR model, a spontaneously hypertensive rat. It's an animal that exists in different strains with different disease susceptibilities, all occurring on the background of hypertension. This animal was produced by selective inbreeding from outbred Wistar rats on the trait of elevated blood pressure, and all animals that are called SHR are derived from this fundamental parental mating between two animals. Those were brother-sister mated for several generations to fix the elevated blood pressure trait, and then inbreeding was continued through a number of different clades. When at about F21, one of those clades on the left-hand side here indicated it was suffering some of the bad consequences of hypertension, in particular stroke. Now, it's hard to breed an animal that's had stroke, so they bred the progeny of animals that had stroke and quite quickly fixed this trait. So we have two different lineages of SHR, one of which is injury prone. That's the SHRA3, also called the SHRSP, stroke prone, as well as other strains. And we're going to be talking about B2, which are all injury resistant. So close your eyes if you don't look ugly pictures. This is what stroke looks like in these animals. You can get major hemorrhages. You can see some on the left and right here. You can also get microbleeds. If I can get my mouse in position, these are subcortical microbleeds here. And on the right-hand side, you can see a major contrast between the brains of affected and unaffected animals. The left hand of those two images is an animal from the stroke-sensitive strain that has major cerebral edema. You can see the cerebral and the cerebellar hemispheres swollen with very poorly defined gaps between them, whereas on the right-hand side, that's what a normal brain looks like. So one of the questions is that we've been hearing about diversity outbred strains and the utility of bringing all of this genetic diversity between strains into power genetic studies. This is sort of the opposite approach, where you take two strains that are quite similar and therefore have limited genetic differences between them and start to seek the underpinning of phenotypic variation due to their similarities and therefore the restricted amount of differences. So the question we asked was, what is the difference and similarity between these two strains that share this ancestry? And as you'll be hearing more about tomorrow, we've been in the last few years doing whole genome de novo assembly using long reads. So we have very accurate and complete assemblies of the A3 and the B2 genomes. And this is a plot of the autosomes, and the lines marked in black represent regions where there's non-identity by ancestry in these two strains. So across the whole genome, about 87% linearly across the genome is identical, 13% difference. So that's a genome-wide variant rate of about 1 in 2,000. So quite similar. And areas where they're extremely similar. For example, here's chromosome 6. There's one major haplotype block on the left-hand side, maybe a small one. And on the right-hand side, this is the most diverse region of the genome between these two strains. It also happens to correspond to the locus that contains the immunoglobulin heavy chain from which antibodies are produced. And we had already noted a difference between these two strains in antibodies. This is an IgG Western blot, and you see this remarkable dimorphism between immunoglobulins in these two strains, where A3 appears to have one missing. And that missing one is the isotype called IgG2b, and you can see in an F2 intercross mapping study, we have mapped the locus that causes the absence of this isotype from this strain, and it maps straight back with a huge log score to the immunoglobulin heavy chain locus. And so there are other differences. G2C also has, and I'm showing data from two different ages in the histogram, this persistent difference in serum IgG levels between these two strains. So because of the isolation of this locus, it was relatively easy to make a congenic strain in which we substituted the IgH from B2 into the SHRA3 background and found, and you'll see some of these measures repeated for the rest of the presentation, but body to brain weight ratio reflecting the brain swelling, the transverse and coronal diameters of the brain reflecting brain swelling, counting of major hemorrhages and microbleeds, and also survival. These animals are given strokes by replacing drinking water at 18 weeks of age with a 1% sodium chloride solution. And quite soon after that, the prone animals start dying off, whereas you can see the protective effect of immunoglobulin substitution here. But we know this is a polygenic trait, and so we've done whole genome sequencing by Illumina a while back to try and look for mutations that might also be distinguishing between these two strains. And we found this fascinating mutation. It's a premature stop codon, but it's in the terminal exon of STIM1. And terminal exons are supposed to have stop codons. We wondered whether we get nonsense-mediated RNA decay out of this or not. And it turns out you don't. We got antibodies to the N-terminus and also to the C-terminus of this protein around western blots. The N-terminus, of course, is preserved in both strains, but you can see the size difference due to the difference in the C-terminus. And the C-terminus antibody doesn't find STIM1 at all. So as we started to study STIM1 a bit more, we realized that human patients that have STIM1 complete insufficiency get a syndrome of immunodeficiency, because it's an immune signaling gene, but they also get antibody-mediated autoimmunity. So antibodies enter the picture again. This is what STIM1 does. It's central to T and B cell calcium signaling, which is the main driving force for activating lymphocytes. And they're coupled to the T and B cell receptors, which initiate depletion of ER stored calcium. But to drive development of immune cells, you have to have a bigger calcium signal, and that comes from the STIM1 acting as a sensor in the ER. And when the ER calcium is depleted, it opens this ORI channel, allowing extracellular calcium to enter the cell and drive these immune responses to activation. STIM1 is on chromosome 1, and it exists right in the middle of this haplotype block that's identical by descent between these two strains. It's presumably a de novo mutation, and it's unique to the SHRA3 strain. But again, this facilitates making congenics because that entire block there contains no other coding genes that have nonsense or non-synonymous variation in them. So we made the congenic strain. And once again, on the left-hand side, I haven't talked about blood pressure. These congenic strains that I'm showing you do not have blood pressure differences until you start salt loading them, and then you produce the injury, and then that affects blood pressure further. But the STIM1 also gives complete protection from stroke, and we have this neurological injury score that's basically a clinical assessment. These animals are very well protected, and likewise the brain weight and body ratio and the physical diameters of the brain. One third gene to tell you about, the first gene, immunoglobulin, is highly structurally variant. JUND looks like it's not structurally variant. It's a gene that lacks introns, but it's been known for a while prior to us that there's a major expression phenotype associated with this gene. So you can see the alignment of our assemblies with the reference assembly, and none of these strains have any polymorphism across them. But yet the SHRB2 and the WKY have abundant expression, whereas across 8 distinct tissues, SHRA3, we see no transcripts whatsoever. And this is related to a promoter polymorphism, we believe. It occurs in cis. And there's one of the nice things about structurally complete genome assemblies is that you can see when you have variations in promoters that are due to things like line and sign elements. And with short reads, because those are abundant throughout the genome, your short reads don't know where to align. And so with these long read assemblies, you can see those, and there's an important one in the proximal promoter of JUND. So the JUND congenic was also made. We again saw the same beneficial effects. So now we have these 3 genes. We know that immunoglobulins are only expressed in B cells, that STIM1 is important for B cell calcium signaling, and JUND is in fact a transcription factor driven by calcium signaling in part, and that acts as a restraint on immune cell proliferation and activation. And that restraint is essentially lost in SHRA3. Well, it seems rather strange to propose that stroke is a B cell mediated disease, but we can test that hypothesis pharmacologically. People who have kidney transplants are often treated with this immunosuppressant called mycophenolate mofetil. It's an immunosuppressant that works by interrupting the nucleoside recycling that lymphocytes alone use as part of their DNA replication process. So if we do this pharmacological replication of B cell, and in this case T cell also suppression, we see a similar benefit. But probably the ultimate test that proves our hypothesis that antibodies in fact create stroke susceptibility in the presence of hypertension is the utilization of the very first rat zinc finger nuclease mediated knockout that Aaron Gertz made. We eventually were able to obtain this animal. It's in a Sprague-Dawley genetic background, but we back-crossed it into SHRA3, so we effectively have an SHRA3 knockout. You can see sequencing here across the IGH locus in the place where that zinc finger nuclease deleted some of the IGH, and you can see on the upper right-hand side that these animals have abundant T cells, the yellow, and over here, but their B cells are completely missing, and their immunoglobulin levels are substantially reduced. Since we have the genome assembly for SHRA3, we can do short-read sequencing and look whether our congenic strains have a lot of hitchhiking material across, and there's just very little evidence of that. These animals are 99% SHRA3, And the major differences between the knockout strain and the SHRE3 is in the IgH locus. So does IgH knockout protect from stroke? And again, I won't spend too much time on the details, but the answer is yes, it clearly is beneficial to these animals. So we've identified 3 genes that influence stroke in SHRSP, the immunoglobulin heavy chain, JUND, and STIM1. 2 of them contain structural variation. I think in the case of JUND, we'll probably be able to attribute that to a sign element that's been inserted in the proximal promoter. IgH is a much more complicated variant. It'll be fun to study. In SHRA3, the IgH locus is about 4 megabases. In B2, it's over 8. So these immunoglobulin and adaptive immune receptor loci are extremely complicated. All 3 of these genes are only co-expressed in B cells, which I think is good evidence that we're on the right biological path. Pharmacological suppression of T and B cell proliferation is stroke protective, and immunoglobulin is clearly involved in the pathogenesis as shown by the knockout. So the question that engages us most at the moment is, what does a pathogenic antibody do to create a stroke in the presence of hypertension? Thank you. Happy to answer any questions.